Hello everyone and welcome to the first video of this channel. Today we're going to be looking at the first topic of the CIE 9700 biology syllabus. And we're going to start with cell structure. So this topic seems pretty minor as compared to the other topics, but in reality it's the key to understanding the bigger concepts of bio. So you can see that many of the future chapters come back and link to this first chapter. So stuff like transport mechanisms, the adaptations of each organelle in different complex functions, it all ties back to cell structure. So let's start off with the difference between magnification and resolution. Magnification, when you're talking about magnification, the first thing that should come into your mind is this formula, which is M equals I over A. So this tells you how you can calculate the magnification of an image. So before I explain what this formula intends to do, I would like to define what magnification is. So magnification is the number of times an object has been enlarged. Now, how this helps is this helps in creating a larger image. Okay? So magnification is the number of times in which an object has been enlarged to create or to obtain a larger image. So we have magnification on this side of our formula, and what the I stands for is the image size, whereas the A stands for the actual size of the object. I will be making a separate video on how we can calculate magnification from specimens, or from when you have your own specimen and you have to draw and you have to figure out the magnification or the image size or the actual size. So I'll be making a separate video detailing that. But for now, let's just keep this simple equation in mind. Now, the problem is students tend to confuse magnification and resolution with each other. When we're talking about resolution, the first thing that should come to your mind is clarity. So let's imagine that this box is an area or like a boundary, and then you have 2 points, okay, in this area. Now, if you have a microscope, which or any piece of technology that enables you to view an image, if you have a device that has a lower resolution, what tends to happen is that these 2 points merge and it forms. So basically it looks something like this. They'll merge together. And what this causes is a blurry image. So the clarity or the crispness of that image has been reduced. However, when you have a higher resolution, these points still are separate. So the device, or in our case, our microscope, is able to distinguish between these 2 points. Therefore, the resulting image that we get is higher in clarity and, in other words, higher in crispness. So the resolution is the microscope's ability to distinguish 2 points, or maybe not two, just points, to distinguish different points found on an image as separate. So this in turn enables us to judge the clarity of the image. Now, one thing that I'd like to make clear is that a higher magnification does not equal a higher resolution. People assume that when you have a larger image, you can see better details. However, you can think of resolution as a limitation of magnification. So no matter how much you zoom into an image, or however much you enlarge the image, it's not guaranteed that the clarity or the resolution would be good. Now, resolution depends on a little something known as a wavelength. So let's imagine that this is a wave, and this part right here is known as a wavelength. Now, for visible light, the range would be 400 to 700 nanometers. So this is specifically for light microscopes, because the source of the source for light microscopes is visible light. So it reflects on to the specimen and then through the lens so that you can see the specimen in the microscope clearly. Now, in order to properly see a specimen, the specimen must hit the wave. Therefore, if you have a wave and it fails to hit the specimen, then you simply cannot see the specimen from the microscope. Therefore, it is said that the smaller the wavelength, the easier it is for it to hit the specimen, and therefore you can see the image clearly. When you're comparing electrons to visible light, you can notice that it uses a smaller wavelength as compared to visible light. Therefore, electron microscopes tend to be higher in resolution as compared to your normal light microscopes that you use in your school. In other words, the ability or the resolving power, okay, keep this in mind, the resolving power of electron microscopes are higher as compared to light microscopes. Therefore, you can see your specimen in much better clarity. So here I have with me a diagram of a animal cell. So in order to understand the differences between a plant cell versus animal cell versus a prokaryotic cell, It's better to understand the differences between the diagrams that we're going to label together. So let's start with the structure right here. This is a cell surface membrane. The cell surface membrane of cells is made up of a structure known as a phospholipid bilayer. Now, what the phospholipid bilayer is it looks a little something like this. We will be exploring this in better detail in cell surface and cell membranes and transport, but a phospholipid bilayer looks something like this with a hydrophobic core from their hydrophobic tails. So this is a tail, which are so these tails are hydrophobic, water repellent, whereas these heads, these are hydrophilic, which means that they attract water. We'll be looking into it in more detail in a model known as the fluid mosaic model. But for now, we just need to know that the cell surface membrane is made up of a phospholipid bilayer. Now, how this helps is that this helps the cell uh, in selective permeability. So the cell doesn't really let everything in the extracellular environment to enter the cell. It only allows some things to come inside of the cell, and it also allows for only certain materials to leave the cell. So, in simpler terms, the cell surface membrane controls the entry-exit mechanism of materials both into and out of the cell. Now, the next structure that we are going to be looking at is the nucleus. There are some organelles that are missing from this environment, from this diagram, so I'm just going to draw those in. Okay? So let us look at the nucleus. This structure right here, this is a nucleus. So what the nucleus does is it's basically the cell's control system. It contains genetic material in the form of DNA. Now this membrane-like structure over here, this, the outer layer of the nucleus, is known as a nuclear envelope. So the nuclear envelope is a double membrane. So one here, then one here. It's a double membrane structure, and it contains small pores within, or small openings, very minute openings, spread across the nuclear membrane as nuclear pores. And what it does is it allows for materials to go out of the nucleus. The nucleolus, which is this circular structure that I've drawn over here, this is a nucleolus. The nucleolus is responsible for making a substance known as rRNA. What this does is that it produces It's basically involved in ribosome synthesis. So the nucleolus makes ribosomal subunits. Let me just quickly write that here for you. So this makes rRNA and ribosomal subunits, which are responsible for creating ribosomes. So the rRNA and ribosomal subunits exit through the nuclear envelope via the nuclear pores. The next organelle that we will be looking into is the rough endoplasmic reticulum, which is this structure right here. I will be abbreviating it into RER, but the examination will not accept RER. You have to write the full name, rough endoplasmic reticulum. So the RER basically is the surface is covered in ribosomes. So these dots over here that you can see, these little dots, this is what causes a quote unquote rough surface. This is the rough endoplasmic reticulum. Because we have ribosomes on the surface of the endoplasmic reticulum, the process it's primarily involved in is protein synthesis, and these proteins are then transported through the Golgi body from here. The structure that you see right here, this is the SER, or the smooth endoplasmic reticulum. They call it smooth due to the lack of ribosomes, but what this does is it's involved in lipid synthesis or steroid synthesis. It's involved in lipid and steroid synthesis, whereas the RER is responsible for protein synthesis due to the presence of ribosomes. Now, I would like to draw a structure. So let me just, so it's a structure that looks like this. This structure right here, this Wi-Fi symbol looking structure, this is the Golgi body or the Golgi complex. The CIE will basically accept the terms Golgi body, Golgi complex, Golgi apparatus, right? So what this is responsible in doing is the modification, pack, packaging, and sorting proteins and lipids. And you can guess where these come from. These come from the RER from here, and then the smooth endoplasmic reticulum right here. What it also does is produce vesicles or lysosomes, which both are involved in packaging substances within itself, which I'll get to later. But let's look at vesicles first. So these proteins or lipids will be found in these membranes, which I will be drawing over here. And then it will come to the cell surface membrane, and then the vesicle dissolves. And then what it does is it leads to the release of whatever this vesicle contains to the extracellular environment. We will be discussing lysosomes in a while, but let's move on to mitochondria. Mitochondria right here is involved in ATP synthesis. ATP synthesis is responsible for the release of energy. Unless ATP synthesis occurs, the cell will lack the amount of energy that it needs to perform its daily tasks to maintain its proper functioning. So the mitochondria is a double membrane structure, as you can see right here, and then you can see right here. It's double membrane, and it has its own circular DNA. So it means that it can code for some protein. Here we have ribosomes, these little dots that we have, which are also found on the rough endoplasmic reticulum, we have them in the cytoplasm as well. So ribosomes are involved in protein synthesis, and there are two types of ribosomes. We have 80S and 70S ribosomes. These tend to be the larger variety, and these are smaller. You can find 80S ribosomes in eukaryotic cells, and 70S ribosomes are found in mitochondria, chloroplasts, and prokaryotic cells. Next, let us look at these. This is let me just move that from here. All right. This right here, the structure, this is known as a lysosome. Now this packages something known as hydrolytic enzymes. Hydrolytic enzymes are a type of digestive enzymes that help in breaking down worn out organelles or waste products or dead organelles. Now, what if this was a plant cell? What could we see extra? If this was a plant cell, we'd have we'd have this extra layer right here. Right above the cell surface membrane. So this extra layer right outside. This extra layer outside is known as a cell wall. Now, plant cells are the major type of eukaryotic cell with a cell wall, which we will be primarily focusing on. So, the cell wall of plants are made up of a substance known as cellulose, which we will be looking into more detail on its formation and of its nature and of its breakdown process in the next chapter, biological molecules. So the cell wall is involved in structural support. It gives the cell structural support. That's what helps a plant cell maintain its characteristic shape. And what it also does, just note this down right here real quick. So it's made up of cellulose, and it helps in structural support. What it also does is it allows the plant cell to maintain its turgor pressure, or basically the pressure within the cell, so that it doesn't burst or it doesn't have a pressure so low that it starts to turn flaccid. Now there is an organelle which is already present here. This is the chloroplast. The chloroplast, much like the ribosome, has two nuclei. Sorry, it has two membrane. My bad. Two membranes, and it has also its own DNA, and these are sacs that contain the pigment chlorophyll. Right. Now, considering that we've turned this diagram into quite an ugly plant cell, let us finish off this diagram with one final structure. The final structure that I'd like to draw, because this diagram lacks it, is where do I draw this? Okay, let's draw a separate plant cell, and then I will. So imagine that this is a cytoplasm where all the organelles are in. There is a sac-like structure in plant cells that are large and covers a lot of the space in a plant cell. This structure is known as a vacuole. A vacuole is a sac that contains a mixture of salt, sugars, and pigments, and in some cases, toxins as well. Now, this vacuole has its own membrane-like structure over here known as a tonoplast. And it's simply just the membrane. It allows for basically transport of these items inside the vacuole. Now, in some cells, which are basically too big So if we have a cell that is too big, let me just draw a random cell over here. So cells that are too big have a very large area to cover. Therefore, the efficacy or how efficiently they absorb nutrients could be reduced. Therefore, they will have these structures known as microvilli, small finger-like protrusions across. And what this does is it increases the cell's surface area of absorption. Right. So this is it for the diagram part. Now, let's compare eukaryotic cells to prokaryotic cells. Now, let us compare the differences between our prokaryotic cell right here, which is a bacteria cell, and our animal slash plant cell, which we've converted it into. So we see that there is a huge difference in the amount of details that we have over here. We do see some structures that we haven't seen in the animal cell, such as this tail-like structure known as the flagellum. The flagellum is a whip-like structure which allows the locomotion or the movement of a cell. Here, these hair-like structures, it's not to be confused with cilia or microvilli. This is a pilus, and it's involved in transferring materials between bacterium to bacterium. Here, we can see that we have the cell wall, something common. However, the composition of this cell wall is different. In plants, we could see that there was a cell wall made up of, guessed it, cellulose. Here, however, the difference is that even though it possesses a cell wall, this cell wall is made up of a substance known as peptidoglycan. Or in other words, murine. But I assume CIE prefers peptidoglycan over murine. So stick to peptidoglycan. Here we have our ribosomes. Ribosomes are involved in, again, protein synthesis. But the difference is that these are 70S ribosomes, so the smaller variety. We have our cytoplasm, as usual. And then here we have our capsule. So basically, this is an extra layer found on top of the cell wall. So when we're looking at a typical, oh, and then we have our circular DNA. Right. So coming back to the topic of prokaryotes, typically a bacterium cell, we can note the following. So what did we observe? We observed that it has a cell wall, but made of peptidoglycan. We also notice that they have circular DNA. They have 70S ribosomes, and they also have a lack, or they do not have, membrane-bound organelles. Now, what they mean by membrane-bound organelles is that basically they don't have organelles that have a double membrane, such as mitochondria or chloroplasts. Now, another fact that I like to throw in here is that prokaryotic cells tend to be unicellular and are much smaller than our usual eukaryotic cells. Eukaryotic cells tend to be larger in size due to the sheer complexity of their structures. However, since prokaryotes tend to be more simplistic, they are in turn much smaller, so they have a size of 1 to 5 micrometers, and this is in terms of their diameters. We're almost done with this chapter, and the final part of this chapter is understanding what a virus is. So, a virus, well, there's an ongoing debate about viruses. So, some scientists are debating that they are cellular organisms. Some are debating that they're acellular, but the theory with most empirical proof at the moment is that viruses are acellular. Okay? Now, let us look at the structure of a virus. Here, this snake-like thing over here, this is the nucleic acid core found right in the center. The nucleic acid core is a structure which can only exist as either DNA or RNA. A virus cannot possess a nucleic acid core of a combination of DNA and RNA. Now, this geometric structure right here, this is a capsid, and this is a protein capsid, which keeps the genetic information or the nucleic acid core protected. Finally, the red part over here, this is known as a phospholipid envelope. And only some of the viruses possess a phospholipid envelope. So these are the three major things that you'll have to keep in mind when you're looking at viruses, and especially the fact that they're acellular. People tend to confuse the structures, and they tend to assume that viruses are in fact cellular when they're not. I would also like to add on to the fact that viruses are dormant, or they are not active unless and until they are within a host's body, which where they can in turn infect the other cells that are healthy by injecting the cells or by transferring their DNA or their genetic information, like their RNA, into the other surrounding cells. Thank you so much for watching. If you guys have any questions, please don't hesitate to drop them in the comment section down below.